But we still have a problem. What happens when the axon wants to communicate with another cell, such as another neuron or a muscle cell or even an endocrine cell or something like that? For that process, typically another mechanism is used. The second part of neurotransmission goes by a variety of names. We'll just say synaptic potentials. Synaptic potentials are synapses. So there are two important components of neurotransmission: the action potential and the synapse or synaptic potential. So today we're going to talk about some of the vagaries of synapses, and you can sort of see an idealized synapse here. Again, the projector sort of obscures our vision. Let's pick the wrong thing there. Now, we're mostly concerned with what are called, in this discussion, with what are called chemical synapses. We've already talked about electrical synapses. What are electrical synapses? We had three names for the same thing.
Membrane potential?
No, remember the electrical synapse is a membrane protein, but what type of membrane protein is it? A gap junction. Yeah, it's a gap junction. We also call it a connexon. The three terms are the same thing. Electrical synapses, those gap junctions, are direct cytoplasmic bridges between adjacent cells. They act very rapidly. They're bidirectional because the depolarization in one cell can be rapidly communicated to the adjacent cell in either direction. These, as we indicated, are present in cardiac muscle. They're present in some types of smooth muscle, and indeed, we've discovered even present in the central nervous system to some extent. Now, by contrast, the chemical synapses, which are these, and we've already looked at them, and we looked at the presynaptic axon terminal. Then there's a space called the synaptic cleft, and then you're going to have the postsynaptic dendrite or cell body. And so we call that whole assemblage, and some other things we'll be talking about today, the synapse. Now, synapses differ from gap junctions in a variety of ways. They obviously are not direct cytoplasmic bridges. Rather, they are more or less unidirectional excitations of the presynaptic cell with the postsynaptic cell. I have to be careful with unidirectional. That's what you see in all the textbooks. As you might imagine, recently it has been discovered that not all synapses are one way, but that won't affect our discussion in here. They're much slower, as you might imagine, because I think, as Bob pointed out the other day, the mechanism by which the signal leaves the presynaptic neuron and passes to the postsynaptic neuron is simple diffusion. So it's, again, quite remarkable. But fortunately, the distance of that diffusion is quite small. Although notice it's larger than that of an electrical synapse, on the order of 30 to 50 nanometers. So synapses have a bit of a delay, about typically a few milliseconds, and you can see it's just by way of comparison, it's about on the order of the neuronal refractory period, right? The same magnitude at least. So that gives you some idea. So let's look at these in a little more detail. And this is sort of your archetypical synapse portrayed on the left here. And we want to make sure we understand this thoroughly, because this is fundamental to our understanding of not only of the nervous system, both central and peripheral, but our understanding of the autonomic nervous system, the neuromuscular junction, and so on. So in this illustration, what we have is the presynaptic cell. I guess it's aquamarine or whatever color that is there. The blue here, I guess, is our cell membrane. And then you can see descending the cell membrane, descending in this illustration, the red rectangle there is an action potential. So that action potential is moving down the axon terminal toward the synaptic cleft. And this white area then is our synaptic cleft. Now, as that action potential, this migrating wave of depolarization, arrives at the axon terminal, this is sometimes called a bouton. The French named this, so they call it a button or bouton. And this, we're just going to call it an axon terminal. As it arrives at the synaptic cleft, several things happen. The details aren't terribly important right now. But the action potential triggers a voltage-gated channel to open. But notice this is a different voltage-gated channel. We haven't looked at this one before. This is a voltage-gated calcium channel. Calcium is more abundant inside or outside the cell? Outside. So when that channel opens, there's going to be an influx of calcium. And one of the fundamental principles of the physiology course you probably already know, I think I've already stated this several times, is that as intracytosolic calcium concentration goes up in a cell, that's a very powerful signal for the cell. It's kind of like a light switch. It's the fundamental mechanism, for whatever reason, that tells a cell to do something. All right, so basically an increase in calcium ion concentration inside a cell is going to cause that cell to do something, change its behavior in some fundamental way. Now in this case, the way that the response of the cell is something called calcium-dependent exocytosis of neurotransmitter. Calcium-dependent exocytosis of neurotransmitter. Cell could do a number of other things in addition to this, but that's the most important. So we won't get into all this, the second messenger signaling involved in the deployment of neurotransmitter, but notice what happens. Action potential arrives at the synaptic terminal, the voltage-gated calcium channel opens, calcium rushes into the cell via the calcium channel, causing once again, you can see the synaptic vesicles here. These probably came from the Golgi apparatus. And now, as we talked about in our discussion of exocytosis, they merge with the synapse membrane, releasing their contents, in this case the little red dots here, which are some type of neurotransmitter. And that neurotransmitter can be, and we're going to talk about the various chemical classes of neurotransmitter in just a moment, but it's basically just a small amine, could be a peptide. And it's going to be released into the synaptic cleft where it diffuses in all directions. It has some parallels down here. Some types of synapses have little enzymes that will gobble it up. But for now, just understand that these red things are diffusing in all directions. And if circumstances permit, if the concentration of neurotransmitter is sufficiently high, they can bind to postsynaptic, again, presynaptic, postsynaptic membrane receptors, and thereby induce or elicit some type of postsynaptic response. So don't confuse the presynaptic events with the postsynaptic events. Right now, we're only concerned with what's happening, at least in this illustration, with what's happening on the presynaptic side. And this could be, for example, a ligand-gated receptor. Yes, sir?
You said if the concentration is sufficiently high enough, it will bind. So if it's not high enough, it won't bind at all, or it just won't cause a...
Yeah, you can imagine we're only looking at one receptor. Here in reality, there are going to be thousands of these and thousands of these, so it's just a statistical probability. So yes, it's theoretically possible. Well, in fact, it's not theoretically possible. It is in fact the case that virtually all presynaptic cells are releasing tonic amounts of neurotransmitter. What do I mean by tonic amounts? That's a term you should be familiar with. It's analogous to constitutive. What was constitutive? Normal, yeah. So this is true for it's most evident in the eye, but it's also true of the neuromuscular junction. So these presynaptic cells are always releasing tiny amounts, tonic amounts, and we could talk about that sometimes, why that would be physiologically desirable. But if the concentration is insufficient for these red dots to make it through the various enzymes or other impediments, they might just diffuse away. In fact, that's happening in your brain all the time. But it's very much the case that if the concentration is insufficient, the signaling, any response of the postsynaptic cell would be negligible.
Is the calcium influx like I would say, let me see how to word it, but the sodium essentially is positive, and that's what kind of triggers the depolarization. When it comes to maybe even being more positive, does it also trigger like more calcium channels to open once they start coming in?
So your question is is the actual signal here a depolarization event, or is there something else going
Well,
on?
does it also have the depolarization?
Yes, it does assist the depolarization, but the calcium ion concentration gradient is so much lower than the sodium. It's, in most cases, not significant. In a few cases it is, and for example, in heart muscle and sweet muscle. In this case, and we'll defer this, the signaling mechanism is much more complicated than simple depolarization, but we'll talk about that later. And it's not really important to our discussion. Depolarization is certainly going on here, but it's not the primary mechanism of this process. There's a mechanism some of you probably studied called second messenger signal transduction. And again, it's this calcium acting as a second messenger, taking information from the membrane to the interior. That's really the signal. The calcium is acting as a little, I don't know, a little megaphone or something, a little transmitter. So that's basically it. This is the mechanism for events at the neuromuscular junction, most neuron-neuron cell communication and so on. But it again also affords us our first opportunity to see how important calcium can be. Calcium is going to come up again and again in the remainder of the course. Now, this is a ligand-gated receptor, as we saw in the discussion yesterday. This receptor, if it's a receptor channel, could be depolarizing or hyperpolarizing. Can you give me an example of a hyperpolarizing receptor? You know that. GABA, exactly right, GABA. So if it's a chloride channel, that would be a GABA channel, or it could be a glutamatergic channel. We'll go over the types of channels in just a moment. Any questions about the general sequence of events at the synapse? We will find that it's true in both the nervous system and the endocrine system that the post-synaptic response, the physiological response of neurotransmission or endocrine communication, is determined primarily by the receptor, not the neurotransmitter. So make sure you appreciate that the same neurotransmitter or the same hormone can have different physiological results, different physiological consequences, depending upon the receptor to which it binds. Now, perhaps this will go some way to addressing Louis' question. For simplicity, in here, and I fully acknowledge this is a dramatic oversimplification, but this is the way we're going to approach it, at least initially. We're going to say that there are two fundamental classes of post-synaptic response to neurotransmitter binding. We're going to say there are two fundamental classes, or two fundamental ways in which post-synaptic cells can respond to binding of neurotransmitters. And we're going to use very simple terms to classify them, to describe them. Number one, we can call these FAST. We can just call these fast synaptic potentials. And what happens in class number one here, in the course of the fast synaptic potentials, is the neurotransmitter binds a ligand-gated channel, and zippity-doo-dah, there's immediate depolarization or hyperpolarization of the postsynaptic cell membrane. Right? So if binding of the neurotransmitter causes a sodium ion channel to open, what's going to be the result? Depolarization. So that's one possible outcome. We also mentioned GABA. There could also be more or less instantaneous hyperpolarization too. But class number one, these are basically just ligand-gated ion channels, and the biological or biochemical response is extremely rapid. The other class, we have the animation here we'll look at. The other class is this second messenger cascade. And so in general, and unfortunately, I can't give you a precise characterization of what happens here, but in general, it'll be something like this. The presynaptic cell releases the neurotransmitter. The neurotransmitter diffuses across the synaptic cleft, binds to a receptor, and at that point, the shape, the conformation of the postsynaptic receptor changes, and that will cause a cascade, a chain of intracellular chemical reactions. Some of those will occur at the membrane, postsynaptic cell membrane, and some of those will occur deeper inside the cell. Some of these can be very short. The sequence of chemical reactions, it may only involve 4 or 5 steps. Some of them, for example, we encounter with growth factors and trophic factors and so on, can be 40 to 100 steps in length. They just go on and on and on. Yeah.
So these are intracellular chemical reactions?
I'd say intracellular, but be sure you understand when I say or when we agree that it's intracellular. In some cases, a large number of these reactions can occur at the cell membrane. And then from there, and we'll talk about that in more detail as we get. So the way, as you might guess, we're going to call these slow. So these are slow second messenger signal reactions. It's going to be a while before the results of that neurotransmitter binding receptor are going to show up in the postsynaptic cell. And then that postsynaptic cell, when all is said and done, could do any number of things. It could also just depolarize or hyperpolarize the membrane, just take a lot longer getting there. But usually something else goes on. Usually it's a more sophisticated process. And we'll talk about some of the other corollary processes here, but for now, just understand this general dichotomy. Because we're going to find that certain chemical classes of neurotransmitter tend to do one or the other. And so it helps us understand their mechanism of action if we know the chemical class.
When a signal molecule, such as epinephrine, binds to a cell surface receptor protein, it activates a G protein on the inside of the cell. The G protein then stimulates adenylyl cyclase to produce large amounts of cyclic AMP within the cell. The cyclic AMP then binds to and activates a target protein such as alpha kinase, which adds phosphates to specific proteins in the cell. The effect of this phosphorylation depends on the identity of the cell and the proteins that are phosphorylated. Calcium ions.
Sorry, I'm going to stop there because that's complicated enough. But as we go back, let's go back here. There are a few points that are quite evident here I want you to pay attention to. So as you look at the animation, what's the red thing up there? What's the red sphere? That's going to be the neurotransmitter. So the neurotransmitter is then going to bind to that receptor. In this case, this is a G protein receptor. This is going to be a G protein receptor. The neurotransmitter binds to a G protein receptor. And the purple thing, which is the G protein receptor, is not the G protein. That's, oops, I can't write on it. The G protein is this thing right here. That's the G protein. So neurotransmitter, G protein receptor, G protein. All right? You're going to learn this later. You don't have to memorize all the details now, but you're going to learn this pathway thoroughly later while I give up on that. Here we go. And so adenylyl cyclase, just note that that is a membrane enzyme. So the G protein activates the membrane enzyme. You can see how we could spend all day going through this. The G protein activates the membrane enzyme. The name isn't important right now. But what that membrane enzyme does, in part, most important right now, it converts ATP. What is ATP? It's the fire, it's the energy, it's the battery, it's the currency of the cell. It's going to work. What does it stand for? Adenosine triphosphate. It's going to convert that into ADP and AMP, cyclic AMP, cyclic adenosine monophosphate. So it's just going to break it down. That's what that enzyme does. It's an hydrolyte. It's an ATPase. And those purple, well, there's no purple thing. I guess that's purple. These things are the cyclic AMPs. All right. And then, as you remember, these activate an alpha kinase. Inside your cell, these kinases are very, very important. This whole thing culminates in what's called a phosphorylation reaction. Phosphorylation. And I just said that ATP is kind of the energy, the currency of the cell. That's not quite true. It's really phosphates, PO4, phosphates, that are the energy and the currency of the cell. Just ATP has 3 of them, nobody else does. But what a kinase does is it takes that phosphate and moves it from one molecule to another, usually from one protein to another, and in so doing activates one protein versus another. So what we have then is just basically a cascade of energy transduction. It's like basically giving somebody a $10 bill, another $10 bill, and so on. We're just giving, and how this ends up, don't know. I can't tell you. Any number of things could happen, but it could end up with just an ion channel somewhere else in the membrane opening and causing depolarization. Or it could cause muscle contraction. It could cause, what do we see? How does, what happens in a presynaptic cell when the voltage-gated channel opens? In a presynaptic neuron, what happens when a voltage-gated calcium rushes in and causes what? Exocytosis. So the consequence of this could be contraction, it could be exocytosis of some secretory product. So don't really know all that, but you can see why we call this slow, right? Long chain, long cascade of reactions. And so that's my distinction here. As you look at the illustration down here, here's our neurotransmitter. It's acetylcholine. We'll be talking about acetylcholine in a moment. How would you describe this pathway that you're looking at in this illustration? Is this fast or slow? Yeah, this would probably be called slow, but you can see it's right there on the margin. What's happening here? We have the neurotransmitter binding a G protein receptor. Here's our G protein. The G protein breaks it, it dissociates, part of it leaves and travels across the membrane to this thing. Now what is this? Can you see that right there? Yeah, it's a potassium channel. What's going to be the effect of opening that potassium channel? All right, the potassium leaves. Does that depolarize or hyperpolarize the cell? Yeah, this would be a mechanism of hyperpolarization. So, yeah, but technically this is slow. Even though there's only a few steps, this would be a slow response.
Something hyperpolarizes involves something less excitable.
Well, don't see, so hyperpolarization just means the membrane potential is becoming more negative. That's all that means. We're moving away from zero. So a neuronal potential is normally what? What is a typical norm that I've asked you to memorize? I mean, negative 70, negative 75. So if we hyperpolarize that neuron, which way are we going? Towards zero or away from zero?
Away from zero.
Away from zero. So it's more negative. That's all that means. And is it clear to you why then potassium would hyperpolarize the membrane? What is potassium is positive or negative?
Positive.
All right. And it's leaving, right? And as it leaves, it's carrying positivity with it, making the interior of the cell more negative. So is that your question? Yes.
So when it inhibits the membrane.
Inhibits the membrane. Oh, I see what you're saying. You're talking about an inhibitory receptor. Yes, correct. If you're talking about potential, yes. I was thinking in just very simple terms, but you're right. In general, there are two. We've talked about two ways to cause an inhibitory postsynaptic potential. What are the two general ions associated with inhibitory postsynaptic potentials? GABA, which is a chloride channel, and potassium, which is a potassium channel. Is that clear? We say that both of those, they work by different mechanisms, but they both do the same thing. So, did you still have a question about the?
I just saw the term when we were talking about hyperkalemia.
Hypokalemia?
We were talking about it being hyperpolarized. It also said it makes the cells less excitable.
Yes.
And
So
less excitable is kind of what I was.
yeah. So you see me doing this the rest of the semester. Remember, what did I mean by when I did this? So this is resting membrane potential, and this is threshold potential. So let's say this is normal in negative 75, and this is normal in negative 55. All right. If I make the RMP more negative, which hand am I moving? My right hand or my left hand? So I'm moving this way. All right. So as I move my right hand farther from the threshold, you would agree that it's going to take more of a stimulus.
Yeah.
That's all that means.
Oh, okay.
So I don't think there are any other points that I need to make here. I'm going to talk about the various classes of neurotransmitter and how we can, again, sort of allocate them to one of the other of these categories. The most common example of a fast receptor would be the nicotinic receptor. We're going to look at that in just a moment. But that causes immediate depolarization of the cell membrane and is present in both autonomic synapses and in the neuromuscular junction. So it's very important. You might be thinking about what would happen to a patient with acute nicotinic poisoning, if that's true, if it involves the autonomic nervous system and the neuromuscular system. Slow synaptic potentials, these are chiefly amines, chiefly catecholamines and so on, so we'll talk about some of these as well. Now this is a table of neurotransmitters, and I wouldn't say these are the classes, but these are the major neurotransmitters that you need to know. And there's good news and bad news with this table. And believe it or not, I've actually tried to simplify things with this. So over here we have the neurotransmitter. You need to be able to say something, describe in some fashion each of these. All right. So what do you need to be able to tell me on the test about each of these neurotransmitters? All right. Number one, you need to know the proper adjective that is used to describe that neurotransmitter. So, for example, although you will hear, I just see some atrocious news reporting, science news reporting. Oh, I saw one the other day on something that was just horribly incorrect. Every paragraph had an error. But my point, I don't know why I went off on a tangent there, but although you will sometimes see acetylcholinergic, the proper adjective to describe the neurotransmitter acetylcholine is cholinergic. You can see adrenergic, and by the way, if I had my way, we would have a pronunciation test. There would be an oral component to this test, because you need to be able to pronounce these terms as well as recognize them. Dopamine is fairly simple, dopaminergic, serotonergic. We don't really use this term anymore, but that's for somehow the adjective has been retained. We'll talk about that. Notice that the adjective for glutamate is glutamatergic, not glutamatergic. It's glutamatergic. Glutamatergic refers to a different amino acid. Isn't this convenient? For GABA, it's just GABAtergic. We don't have to spell it all out. Glycinergic. Sometimes you'll see peptidanergic. I don't think that's terribly critical. We'll talk about gases. All right. And then in the next column, I have listed a putative chemical class. This is just for convenience, because acetylcholine is an amine, but we put it in its own class because it's so important. So we have the amines, all right? And then we have the amino acids, a distinction we'll dispense with as we get to the endocrine system. And then we have the peptides and gases. So basically we're talking about cholinergics, amines, amino acids, proteins or peptides, and gases. The fourth column is the synthetic enzyme associated with that neurotransmitter. In other words, what enzyme catalyzes synthesis of the neurotransmitter. You need to know this synthetic enzyme for acetylcholine. You need to know that one. Oh, yeah, that's right. Just know that the catecholamines, you don't need to, that pathway is a little complicated, but know that catecholamines come from tyrosine. Serotonin comes from tryptophan, so those are just hydroxylases that make that chemical conversion. You don't need to know this or this, so you should know the synthetic enzyme for acetylcholine, and know that the amines come from tyrosine, with the exception of 5-HT or serotonin, and that comes from tryptophan. You probably heard about the turkey making you go to sleep at Thanksgiving, and it turns out to be public foolishness. I'm sorry. Now, in addition to the enzyme associated with synthesis, you should know a few of the enzymes associated with breaking down, degrading that neurotransmitter. So for acetylcholine, it is acetylcholinesterase. Acetylcholinesterase. For the amines, we have two of them we need to pay attention to, MAO and COMT. MAO is monoamine oxidase, and COMT is catechol-O-methyltransferase. You don't have to worry about that, but just recognize these two as enzymes that break down the amines, and there they are over there on the left again. And you don't have to worry about these. You should already know that peptides are broken down by proteases. What would break down a lipid? A lipase. Yeah. So. All right, this column here on receptors is very, very important. This is absolutely critical. So what you'll notice, for example, is that cholinergic neurotransmitters have two classes of receptors, nicotinic and muscarinic. And what you notice is the important difference between the two. One is fast, one is slow. So for acetylcholine, there are two fundamental classes of neurotransmitter, the nicotinic and the muscarinic. The nicotinic is fast, causing immediate depolarization of the cell. The muscarinic, any number of things we'll talk about that later. Anytime you see reference to alpha or beta receptors, you're talking about adrenergic receptors. So that's going to be very important in our discussion of the autonomic nervous system and sympathetic activation. But alpha or beta applies to the adrenergics. What do we mean? Can somebody give me an example of an adrenergic neurotransmitter? Adrenal. Yeah, epinephrine and norepinephrine, or adrenaline and noradrenaline. So those are the two adrenergics. And things are a little simple here. Those are both slow. So the adrenergics are always slow, utilizing usually a G protein signaling pathway. Dopamine, dopaminergic 1, dopaminergic 2, and so on, slow. Serotonin receptor 5-HT, these are all slow. So that's the rule. That's one rule I want you to pay attention to. Amines, all the amines, utilize the slow signaling pathway. Again, acetylcholine is in its own little class there. That takes us down to the amino acids. We have glutamate, GABA, glycine, and what do you notice about them?
They're all fast.
They're all fast. They're all fast. They alter membrane potential. Yes?
For the dopamine, is the D1, D2 the reason dopamine affects different cardiovascular amino areas?
Exactly, and we're not even getting into that now, but you're exactly right. For each of these classes of receptor, there are multiple isoforms. There are multiple subclasses, and what is the rule that we articulated earlier? It's not the neurotransmitter that determines the physiological response. It's the receptor. You're exactly right. There are many types of receptor for each of these.
So for serotonin,
Good, that's what I was coming to. I think I see your question. What is it?
I'm trying to read it. Is that
I know. Okay, good, yes. So there is one exception to the serotonergic receptors. The 5-HT3 is fast. All the others are slow. 5-HT3 is an ionophore. All the others are slow. And we'll come back to that later as we talk about serotonin. Basically, I'm just taking an entire 2 hours of lectures and putting it in one table. So it helps all of us. And then don't worry too much about this column, excitatory or inhibitory. I think you can probably deduce whether a whether a receptor is excitatory or inhibitory from other factors. And so, obviously, remember our general rule: it's not the neurotransmitter that determines the response; it's the receptor. So the receptor is really going to determine whether inhibition or excitation occurs. And then, as we proceed, on the final column over here, we'll talk about antagonists and agonists. But first of all, what is an antagonist? What do we mean by an antagonist? What? That's fine. Yeah, the antagonist is a blocker. Actually, said the antagonist is a blocker, and I think that's a good way to think about it. What's an agonist?
Initiator.
Huh?
Initiator.
Okay, basically a pharmacological agonist is a drug or molecule that excites or causes the normal biological response. By that I mean when that drug binds the receptor, it causes the normal thing to happen. And students sometimes say that an antagonist does the opposite of an agonist. It doesn't. Is that clear? An agonist causes the normal biological response. What does an antagonist do? Just blocks the normal biological response. Inhibits the normal biological response. Doesn't do the opposite. All right, so here we have a column called antagonists, and a few of these you should know. You can see, for example, for nicotinic receptors, curare is an antagonist. So curare would effectively block nicotinic neurotransmission. For the muscarinic, scopolamine and atropine are both antagonists. You should know both of those because that's going to come up later in your clinical. We'll talk a little bit more about it. You've heard of a beta blocker? What's a beta blocker?
I don't know, but I've heard about it.
Beta blocker just blocks the beta adrenergic receptor, a beta blocker. And so the physiological consequences of a beta blocker would depend upon where those receptors are. So we need to talk about that. And the rest of these you don't have to know. I just put them up here for completeness. If the square is empty, you don't have to know that. But know the cholinergic antagonist and the adrenergic terms, which are very simple, alpha and beta blockers, and we'll leave it at that. Any questions about this table? I think it summarizes a lot that we don't have to deal with. So this is just the thing I made earlier. This just to sort of summarize it. I took this slide out and then I put it back in. Fast and slow. So how did we categorize chemically the fast receptors? The amino acids and the cholinergic nicotinic. The amino acids and the cholinergic nicotinic for our purposes. And then the slow, all of the amines along with the cholinergic muscarinic, and then most of the neuropeptides.
That's a little fast.
Let's talk a little bit about acetylcholine. Most of this should be redundant with respect to that table, but acetylcholine is synthesized in part by the activity of that synthetic enzyme up there, choline acetyltransferase. Choline acetyltransferase, oftentimes abbreviated as just CHAT, but it's pronounced CAT.
Can you repeat that? I'm sorry.
Yes, if we go back to the slide here, I wanted you to know from that table the synthetic enzyme, the enzyme that's responsible for catalyzing synthesis of acetylcholine. It's choline acetyltransferase, but the way that's normally abbreviated in the literature is just spelled out, but it's pronounced CAT. Sometimes you see it with a capital H. The catabolic enzyme, the enzyme that breaks down acetylcholanes, and this is going to be very important, you need to know this one thoroughly, is acetylcholinesterase, and you can see the abbreviation name there. A capital C, H capital E, acetylcholinesterase. And it lives right in here. It lives in the synaptic cleft. It's like a little Pac-Man gobbling up whatever it encounters. And it breaks it into choline and acetate. Both of these can be either recycled into the Krebs cycle or the membrane. It becomes phosphatidylcholine or something and be recycled into the membrane. So when the acetylcholinesterase breaks down acetylcholine, the metabolites get rapidly recycled. Acetylcholine can be excitatory or inhibitory. If it's nicotinic, it's always excitatory. You can always say that because it can cause rapid depolarization. That nicotinic receptor is a sodium channel. But what about the muscarinic? It's a G protein cascade. Can't tell you. It could do any number of things. Unless I know the pathway, I can't tell you what it can do. I only know that it's going to involve a lot of steps. So here are the two classes again of receptor, nicotinic and muscarinic. Make sure you're familiar with these. Nicotinic is fast, muscarinic is second messenger. And these are, these are all over the place. These are in your brain, nicotinic receptors in the neuromuscular junction. It's in the autonomic nervous system. We're going to have lots of intimate conversations with nicotinic receptors. Muscarinic receptors also in your brain, also in the autonomic nervous system. So both of these are going to figure prominently in the remainder of the course. And so what are we looking at here? What type of receptor is this? This is a acetylcholine, cholinergic receptor. Which of the cholinergic receptors is this? Yeah, this is the nicotinic. Here's our ion channel. The acetylcholine there, which is portrayed as the lemon and the orange together, binds to the membrane protein, causing the channel to open, so
Are all nicotinic receptors going to be sodium channel receptors?
and there are multiple isoforms of nicotinic receptor, but they're all sodium channels. Yeah, so we'll go over the location a little later. It's basically a cation channel, but because there's so much more sodium than potassium, it's the depolarizing effect that's important. And so, with respect to the discussion earlier, the nicotinic channel would be excitatory or depolarizing. Antagonist, we mentioned curare. Curare is a paralytic. It figures in a number of TV shows and movies whereby the poor victim of the curare poisoning is paralyzed, can't move their muscles, but the afferent nervous system is still functional. So you can see all the manner of horror this can lead to in suspense. So what do I mean? They get this from an alkaloid plant. They put it on the arrows in South America. They shoot animals with it. But if the poor animal happens to be a human, what happens? You can imagine all the plots that could unravel. The person is aware of all the horrible things happening to them as they're being tortured or whatever. Yeah.
So this doesn't affect like autonomic, it only when
Oh, it would, yes, it would. The nicotinic antagonist would also, the curare, I don't want to get into the ANS right now, but it most certainly would be.
I was just curious as to how they can still breathe if it's
They wouldn't indefinitely. That aspect of receptor function does not figure prominently into the plots of these TV serials and TV shows. So, yeah, but you're exactly right. Yeah.
I'm just curious if you have theories as to why we have nicotinic receptors.
So what's my alternative? What would you put in their place? I guess I'm not sure. This is a type of channel that is very rapid, very excitatory. There is a more excitatory channel in the brain, in the CNS, but they both are critical for everything from, let's say, rapid signaling for you to send a signal to your motor function, but also between neurons in your central nervous system, both acetylcholine. In fact, in that previous slide, there was a discussion of acetylcholine and learning. If, like me, you happen to be diagnosed with Alzheimer's disease at some point in life, what's the primary medication they're going to prescribe for you? There's some new medications now, but up until a few years ago, what were the only 3 FDA-approved medications for AD? Well, there were drugs that affected, we'll talk about it more in just a moment, but there were drugs that influenced cholinergic pathways in the brain. Because what you find in Alzheimer's is that there is a depression, especially in the frontal cortex, of cholinergic activity. So I don't know, I couldn't really answer that question because whenever somebody asks a why question, I look for selective pressures. And because I don't know what the relative selective pressures, in terms of evolutionary biology, I don't know what the relative selective pressures would be in that case. I don't know what the alternatives would be. respect to the muscarinic receptors, remember these are G protein receptors. These are present on, we will discuss their abundance later, but certainly certainly present in the autonomic nervous system, and once again in the CNS all over the place. And depending upon the pathway, depending upon the consequences of the G protein pathway, they can be excitatory or inhibitory. We've already established why they happen to be slower. And you should know that there are multiple types of second messenger signaling. So this is only one type, and later we'll talk about this. But muscarinics are G protein second, or utilize the G protein second messenger pathway. And we will, boy, you're going to learn all about. You're going to be thoroughly disgusted by the time you finish our discussion of the autonomic nervous system. But you're going to learn about all of these receptors and how they influence everything from heart function to respiratory function to perspiration. Everything, you just name it, it's involved. Now, in bullet number 5 here, you'll see that the muscarinic receptors, the isoforms, are labeled M1, M2, M3, and M4, and so on. As you look at that list, which you do not have to memorize, but what are the primary differences between the M1, the M3, and so on? What's the difference that you notice? Or their location. So I just want you to know that each of these has a slightly different response curve, but these are distributed, these receptors which determine the physiological response are in very specific locations, in this case in the CNS. So it's the receptor and the location of the receptor that determines the biological response, not the neurotransmitter. And what we're looking at down here, if you're explaining to a 5th grader what this is down here, what would you say? What are we looking at? So what is this? This is a fancy word for what? A ligand or neurotransmitter. In this case, we'll assume it's, by the way, you should be able to give me 2 agonists for the muscarinic receptor right now. Give me 2 agonists for the muscarinic receptor. And not for the muscarinic. That would be one of them for the nicotinic, but you're on the right track. So what would be, I'm not going to tell you what he said because that would give away. What would be 2 agonists for the muscarinic receptor? Acetylcholine, and what else? Muscarine. But what do they do? They bind to the receptor, initiating the normal biological response. So if you look at here, here's our neurotransmitter, here's an agonist. This could be acetylcholine, binds to the G protein receptor. Here's our G protein, and dissociates the beta gamma unit. Well, the beta gamma unit do something. The alpha subunit binds to the adenylyl cyclase, or in this case, the phospholipase C. And we didn't talk about this pathway, but that's going to result in a variety of intracellular changes. Then once again, I just happened to show us, cause what? What's this thing? That's a potassium channel. And getting back, is that going to be inhibition or excitation? Inhibition, right?
No idea.
Hyperpolarization of the memory. Any questions about that? A lot of terms to keep straight here. And you should know these 2 antagonists, scopolamine and atropine. When you go to, there used to be a bottle of it up here. When you go to the eye doctor, I'm glad to make an editorial comment, but again, I'll get sued. When you go to the eye doctor, what do they put inside your eye, and what does it do? They put an atropine type analog. Used to, in the old days, back in the 19th century, there was something called belladonna, which the women would sometimes put in their eyes because it would make the pupils much larger. And today we associate that with something else, but then it was thought to be very beautiful. But in any case, that's because it's acting as an antagonist on those autonomic pathways. Scopolamine has, that's been used sort of. Am I familiar with scopolamine?
Yeah, it's this crazy drug that they use down in Brazil, and it's like
legitimately mind control. Like, people will someone will give it to someone else, and then they will ask you, like, hey man, we're gonna go to your apartment, and you're gonna load all of your stuff up into a truck, and go
Somebody taking notes.
It's the craziest thing. They're like zombies, and then they'll wake up. It's like being goofy, but you're still.
Which I can attest to. That's why I have to make it through the class.
It's the scariest. It's the scariest.
Yes, all of you as an inhabitant, it's an inhibitor. Yeah, it's usually something, usually so, yeah, if you don't have more devious plans, I was excited about that, frankly. But I think both of you are like that. But in terms of, it's a better, it has been used for nefarious purposes. It has been used as a depressant, a CNS depressant, and therefore people can become less aware, less vigilant. For that reason, it's analogous to some of the date rape drugs for that reason. But in this context, I just want you to be aware that it's acting upon both the ANS and the CNS in terms of antagonistic effects. Oh, let's take a break. My goodness, I was getting so excited after I took a break.
Because DNS and all that.
ANS and CNS.